Introduce Ervin. So Ervin did his bachelor's in science in biology in the University of Vienna. Then later he pursued a master's also in molecular biology in the University of Vienna. And since 2022, he has joined the Lund University in Sweden to work with Dr. Philip Pereira, working on a really fantastic project who I have been following a lot. And he's also a scientific communicator, and so he's also interested in sharing science as well. And he has received multiple awards and presenting his work in like 15 places. And I think it's really fantastic that this work is being shared. And I'm really happy to have you here, Erwin.
Thank you so much for this really kind introduction and long introduction. I'm really happy and really, it's my pleasure to be here to presenting to you today our work. As I said, I'm a PhD student in Philip Pereira's lab in Sweden, and I've been working on this project now in the last years during my PhD, and this work has been published quite recently in Science magazine with the title In Vivo Dendritic Cell Reprogramming for Cancer Immunotherapy. And the topic is today about using dendritic cells for therapeutics. So using the reprogramming of dendritic cells for therapeutics. But before I start talking about the project itself, I will just give a brief introduction in the topic. So it's now very known that cancer immunotherapy modalities have revolutionized cancer treatment in the last years, particularly the use of checkpoint inhibitors, since they have been used in various types of cancers and have really led to long-term survival of patients. And we see these really good response rates in melanoma patients, which makes us very happy. However, we still see also a big percentage of patients not responding to checkpoint inhibitors in melanoma, but as well, even more in other types of cancers, which is very unfortunate. And some of the reasons why these patients do not respond is that antigen presentation regulated in tumors, as well as the lack of antigen-presenting cells, high tumor heterogeneity, and one of the biggest issues also for immunotherapy is the immunosuppressive microenvironment, blocking the efficacy. And I think I don't have to convince you that dendritic cells are very important in cancer immunotherapy, but particularly this subset of conventional dendritic cells type 1, in short, CDC1s, are indeed very critical as they can elicit cytotoxic T cell responses against the tumor. And as Pei-Feng before also introduced the migratory and resident DCs, indeed they can adopt different states. So CDC1s can either become migratory DCs, traveling to lymph nodes and activating de novo T cell responses for against the tumor, or they stay actually on the border of the tumor, and from there cross-present tumor antigens to T cells, to CD8 T cells, or present them on MHC2 to CD4 T cells, and also remodel the tumor microenvironment by secreting pro-inflammatory cytokines and chemokines to recruit T cells to the tumor. And these are very crucial functions of CDC1s that, when we look into patients, do correlate with better responses. So we see an increased T cell infiltration when we have higher presence of CDC1s. We see this across different types of tumors, especially when we compare it also to other disease subtypes. And importantly, we see that patients that have more CDC1s in the tumor also respond better to immune checkpoint blockade treatment. So a good synergy. Problem is there's still no available method to generate a pure population of CDC1s to use it for immunotherapy. And in our group, we are very interested in direct reprogramming, since it offers a strategy to generate cell types of interest using the overexpression of transcription factors from an unrelated somatic cell type. So what does that mean? We can use an unrelated somatic cell type, overexpress transcription factors in that cell, and generate another cell type that we need for therapeutic use without passing through a pluripotent intermediate step. And one of the immediate uses of cell reprogramming could be cellular reprogramming in vivo, as you can also directly deliver the transcription factor combinations that are required to reprogram a cell into another cell to the tissue at the disease location to generate the cell of interest. And this has a lot of advantages, as you can generate an off-the-shelf treatment you can generate using a viral vector to deliver these transcription factors. It is immunocompatible since, of course, you're reprogramming the patient's own cells into the cell type of interest. Bears lower tumorigenic risk since you use direct cell reprogramming. And it simplifies a lot the manufacturing. So you don't need ex vivo cell manipulation protocols. You would need to generate viruses. And there are some examples that have been used cellular reprogramming in vivo. For example, there is this approach to reprogram pancreatic cells into beta cells that can produce insulin, or for example, fibroblasts that can be reprogrammed into myocardiocytes that can function in the heart after a heart attack. And so cellular reprogramming has several advantages. However, what you need for an efficient therapy is a good vector. So this is one of the challenges. So you need to identify a good vector to deliver the transcription factors. And so far, adenoviruses, lentivirus, and AAVs have been used the most. And also you need to confirm that the same transcription factor combinations that has been mostly identified in vitro also works in vivo. For example, in neuronal reprogramming, there is transcription factors that have been found that work in vitro well, but do not reprogram the same neuronal cells in vivo. So all this said, we, as a group in 2018, identified PO1, RF8, and BAT3, so very famous transcription factors important for CDC1s to be also to bear the capacity to reprogram unrelated cell types such as fibroblasts into induced CDC1s. And we have published the study in 2018 in Science Immunology, and later also in 2022 reprogramming human fibroblasts into also published in Science Immunology to generate these induced DC1s that do acquire all the DC1 features such as secretion of pro-inflammatory cytokines, chemokines, presenting antigens on MHC2, cross-presenting on MHC1, and importantly, acquiring a transcriptional and phenotypic program of CDC1s. And later, in 2023, so last year, we published as well in Science Immunology that we can not only reprogram fibroblasts, but also cancer cells into so-called tumor antigen-presenting cells. So using a polycistronic lentiviral vector that encodes for PO1, RF8, and BAT3 divided by 2A sequences that will keep the protein separate after translation, we can overexpress this over 9 days, and at day 9, the cells will be completely reprogrammed into CDC1-like cells. And by this, they lose their tumorigenic potential and increase immunogenicity, acquire DC functions, and when we inject them in vivo into tumors, we see longer survival of mice, but more control of tumor growth and antigen-specific T cells expanding. And this, if you want to listen more about this, was already posted on YouTube on Open Box Science, so presented by Olga Zimmermanova, a former postdoc from our lab. So I can just direct you to this YouTube video. But now, since this approach, just talking about the next approach that we are targeted here to bring this to the clinic, we would need to use cancer cells from the patient, reprogram them in vitro, and re-inject them into the patient. And this is not easy. So what we hypothesized is using dendritic cell reprogramming in situ, translating this in vitro reprogramming approach to an entirely in vivo reprogramming approach to directly reprogram cancer cells within the tumor into dendritic cells type 1 and force them to present their antigens and act as dendritic cells type 1. And this study was divided into 3 parts. First, we wanted to assess whether it is feasible to reprogram cancer cells into dendritic cells type 1 within the tumor microenvironment. And we used 4 syngeneic models covering low to high immunogenicity and analyzed immunity and also the immune mechanisms that they are eliciting. And then we translated those findings into the human system using human spheroid and xenograft models. And we reprogrammed them in 3D and also assessed the role of immunosuppression, whether this can impair the acquisition of an immunogenic program. And lastly, we tested this as a gene therapy using different vectors, comparing them, which vector would be the best to generate a gene therapy based on CDC1 reprogramming in vivo and assess the efficacy. So starting with the first part of the study that was a few weeks ago published as first release in Science, we use these melanoma cell lines that are covering immunogenicities from low to high. B16, very common cell line used for melanoma. BRAF, mutated ones that grow very efficiently in bat-free knockout mice, YAM 1.7 and B2905. So to explain you the experimental setup that we are using now throughout the next experiments that I will show you, we used the reprogramming CDC1 reprogramming vector encoding for the free transcription factors, in addition eGFP to track transduction. We used an empty control vector, also encoding for eGFP, just to control for lentiviral mediated immunogenicity. And in addition, we used a macrophage reprogramming vector encoding for PO1 and CBP alpha to compare also whether the CDC1 functions, which are unique for CDC1s, are actually beneficial over the myeloid reprogramming vector, to compare also whether this in vivo translates into better survival. So what we did is we transduced cells in vitro, mixed them with untransduced cells, and implanted in vivo right after transduction to allow the cells to form a tumor, and within the tumor to reprogram into DC1s, and use this as a monotherapy with isotype control antibodies injections, or in combination with anti-PD-1 and anti-CTLA-4. And what we found was very striking. So while the control tumors transduced with eGFP just grew out very fast within 20 days, as these B16 tumors are very aggressive, we saw that PO1 and CBP alpha, as well as PO1, IRF8, and BAT3 induced tumor growth delay. However, we saw that PIB, the combination, even induced individual regressors in the B16 model, which was very exciting to see that the monotherapy already induced strong immunity. But then when we combine this with ICB, so with anti-PD-1 and anti-CTLA-4, we see complete regression in all animals. Also, confirming the findings from clinical studies showing that CDC1s correlate with better response when combined with immune checkpoint inhibitors. So then we looked at the mice and also saw that there was vitiligo on the skin of the animals right above where the tumor was injected, indicating a strong cytotoxic anti-melanoma response. We expanded to other models and saw that in more immunogenic models, such as the YAM 1.7 and B2905, which are, I have to mention as well, still resistant to anti-PD-1 and CTLA-4, we saw complete regression. And these striking results confirmed that we can actually reprogram in vivo and induce anti-tumor immunity. And we looked into the blood of these animals and found that CD8 T cells and CD4 T cells that are specific for melanoma antigens are increased in the blood of animals that were treated with in vivo CDC1 reprogramming. And this already points out towards a systemic response against these tumor antigens. Then we used CDC1 deficient animals, bat-free knockout mice, which have been injected with this BRAF model that only grows in bat-free knockout animals due to the lack of endogenous CDC1s, and induced in vivo reprogramming, and saw that we have indeed as well efficacy here as monotherapy or when combined with anti-PD-1, indicating that in vivo CDC1 reprogramming does not require endogenous CDC1s to act. So we re-challenged the animals to assess whether immunological memory was established. At day 100, only with tumor cells, so there was no treatment involved anymore in the survivors. And YAM 1.7 survivor animals potently rejected the cancer challenge. There was just this unlucky mouse here. And then also the bat-free knockout animals rejected the re-challenge, indicating that once anti-tumor memory is established by CDC1 reprogramming, it becomes also independent of endogenous CDC1s. So we tested whether immunity is systemic in a functional level. And we injected, again, a one-to-one mix with transduced and untransduced melanoma cells. On the left side of the mouse, we injected just untransduced melanoma cells and used the third tumor to control whether the immune response is systemic, but also tumor specific. And for this, we used a different type of tumor. We used the lung carcinoma model. So then what we found was that not only the treated tumor on the right side of the animal, but also on the left side of the animal showed treatment effects, was smaller, and the tumor growth was controlled, while LLC, so the lung model, was not treated, indicating that systemic immunity was indeed induced and it was tumor specific. So next, we looked into the tumors on day 9 after establishment, and while control tumors were typical globular shape of a tumor, of a subcutaneous tumor with immune cells around the tumor, we saw high infiltration of CD45 cells that are marked by this pink color here into the tumors. And I think it is very obvious that we have these really dense CD45 clusters here building up, these structures that we then stain for different immune lymphoid populations, and found that they contain CD19 B cells, CD4 T cells, CD8 T cells, and those TLS-specific stromal cells, indicating that CDC1 reprogramming in vivo induces tertiary lymphoid structure formation. So we performed immunophenotyping by flow cytometry, found again that CD45 cells are increased, so we have more immune cell infiltration into those tumors, B cells, NK cells, and CD4 T cells among those CD45 cells. And interestingly, we did not see really an increase of CD8 T cells at time point day 21 after tumor establishment. But what we saw is a decrease in exhausted CD8 T cells and CD4 T cells, also a decrease in regulatory T cell populations, and an increase in TCF1 positive T cells, which are described to be stem-like T cell populations that give rise to effector T cells and control in a persistent manner tumor growth. The reprogrammed cells, we also looked how long they remain in the tumor. We found them for 9 days in the tumor, and they did not migrate to tumor-draining lymph nodes, and they lack CCR7 expression. So then we further analyzed them by bulk sequencing and found that these reprogramming, that CDC1 reprogramming induced a resident program that is associated with a very immunostimulatory program by the expression of co-stimulatory molecules, chemokines such as CXCL9 and 10, and antigen presentation genes very highly, and did not induce a migratory program. Then we ablated mice from individual lymphoid effector populations, and this was a bit surprising to us, but very interesting that it seemed that CD4 T cells were critical for anti-tumor immunity induced by CDC1 reprogramming. So we performed T cell sequencing on a single cell level and also analyzed the TCRs, and this was done together with our bioinformatician Ilya in our lab. And what we did was what we found was that cells, CD8 T cells in tumors that contained in vivo reprogrammed cancer cells, that CD8 T cell differentiated more towards effector and memory subtypes. You can see this here, and less to exhausted CD8 T cells and to terminally exhausted CD8 T cells. In the CD4 T cell compartment, we found more cytotoxic CD4 T cells, which was again confirming that CD4 T cells are here critical for anti-tumor immunity, and more to TH, so T helper subsets. Looking at the clonal expansion, we saw no clonal expansion. I mean, we saw similar clonal expansion to control tumors here, but mostly in the CD4 T cell compartment, we saw high clonal expansion of CD4 T cells in tumors treated with in vivo reprogramming. And so finishing this part about the mouse and syngenic data and going to the human cancer cells, we also wanted to translate these findings to human cancers, and found that when we compare the in vitro phenotype with the in vivo phenotype induced by reprogramming, is that these glioblastoma models, as well as these melanoma models, when reprogrammed in vivo in immunodeficient animals using xenograft models, acquired in both cases similar efficiencies as CDC1 phenotype. And one difference that we found was that the cells that were reprogrammed in vivo, so surrounded by an environment in an animal, were upregulating CDC1 specific markers such as XCR1, CLEC9A, and CD226, indicating that the in vivo environment increased the acquisition of a CDC1 phenotype, meaning an increased reprogramming fidelity. So next, Malavika and Olga's former postdoc in our lab and research assistant in our lab modeled spheroids to model human cancers and transduced them with PIB and found more expression of CD45 and HLA-DR even in 3D, and then performed this for a broad panel of cancer cell lines that were coming from different types of cancer, indicating that indeed CDC1 reprogramming of different human cancers can progress in 3D. And interestingly, when performing single cell sequencing, they affiliate more to the CDC1 program at earlier time points already at day 3, as well as this tumor APC signature that we created, indicating that reprogramming in 3D is faster and also acquires an immunogenic CDC1 program indicated by this gene set enrichment analysis for the immunogenic gene sets. So then they modeled immunosuppressive tumors using spheroids containing cancer-associated fibroblasts, myeloid-derived suppressor cells, and pericytes, and found that reprogramming into an immunogenic CDC1-like cell is not impaired by the presence of immunosuppressive cells, and also the function. So when we added PBMCs to those spheroids, they acted very efficiently, killed the spheroids. So we found smaller spheroids after the addition of PBMCs that have been not pre-activated with CD3 and CD28, and they secreted pro-inflammatory cytokines and granzymes. So the next part is about the gene therapy, and this was done in close collaboration with Asgard Therapeutics, who is developing this gene therapy to bring it to the patient. And we compared lentiviral, adenoviral, and AAV vectors to inject directly into the tumor the free transcription factors. So when comparing this in 2D and 3D, using spheroids and 2D cultures, using also patient-derived cancers, we found that adenoviral vectors were similar, inducing CDC1 reprogramming as lentiviral vectors with similar efficiencies. So we injected them in vivo into B16 tumors using, again, a mouse model, and found that adenoviral vectors were really efficient in transducing cancer cells in vivo by, we quantified EGFP expression. So combining these two results, high reprogramming efficiency, high transduction efficiency in vivo, we selected adenoviral vectors to move on for a gene therapy approach, injected adenoviral vectors encoding for PO1, RF8, and BAT3 into this sarcoma and melanoma tumors as xenograft models in immunodeficient animals, and found efficient CDC1 reprogramming inside of those tumors. So we selected this gene therapy approach to test the efficacy. And we again moved back to this hard-to-treat tumor, B16, combined injections of adenoviral vectors encoding for PIB together with immune checkpoint inhibitors, and then analyzed tumor growth. Found that 4 out of 8 animals completely regressed in tumor size, remained tumor-free for 100 days, and when re-challenged, stayed tumor-free for another 60 days, so 160 days without any tumors. And then we re-challenged them once again, but this time intravenously, to test for metastatic protection. And what we found is that survivor animals stayed metastasis-free almost, while naive control animals had lungs full with metastatic nodes. So to conclude my presentation today, I would like to just remind you also what we showed. So we can induce in vivo reprogramming of cancer cells into CDC1-like cells by the injection of adenoviral vectors encoding for PO1, RF8, and BAT3. These reprogrammed CDC1-like cells in the tumor are not impaired by immunosuppressive cells and can lead to the formation of tertiary lymphoid structures, T-cell recruitment and activation, and also lead to systemic T cells that are in the circulation and can protect from lung metastasis. In the end, we saw regression with CDC1 reprogramming as monotherapy, but also a significant synergy with anti-PD-1, which leads to abscopal effects, so systemic immunity and immune memory. So with this, I hope that we can soon establish direct reprogramming as a new modality for cancer immunotherapy, and test this hopefully soon in patients. And I would like to acknowledge my supervisor, Felipe Pereira, who gave me the chance to work in this really exciting project. Generally, the whole team of the Pereira lab, who are really smart, brilliant people, very efficient, very helpful, supportive, and of course, all our collaborators, particularly also the team of Asgard Therapeutics. It would not be possible without them and other collaborators around in our center, our funding agencies, and of course today to you, to listening to me today, and hopefully you have a lot of questions for me. Thank you.
Thank you very much, Ervin. So if anyone has a question, please type it in the chat or just unmute yourself. In the meantime, I have a question, Ervin. First of all, congratulations. But I was wondering what happened with the reprogrammed cells after the tumor is like it's eliminated. Like, can you isolate once again in the tissue the reprogrammed cells when the tumor
Yeah.
is gone?
Yeah, exactly. So I can just quickly also go back to the slide. So what we found was that the reprogrammed cells remain in the tumor
Oh,
for approximately 9 days, and then they're gone.
okay.
So dendritic cells are short-lived, and even cancer cells, when we reprogram them into CDC1s, are also short-lived. They remain for 9 days approximately, and then they are gone. They are diminished, probably on the one side killed by T cells. So we see a lot of T cell killing, efficient T cell killing, but also they stop dividing, and at some point are probably apoptotic and die. So what we think, what we hypothesized is that we need them only within the first 9 days in the tumor to
Mhm.
kickstart immunity, and then once the immunity is kickstarted, it's a self-ongoing process.
Okay. And when you compare the like when you compare fibroblast reprogram versus cancer cell reprogram, the cancer cells are more like they have more a potent function than fibroblast reprogram dendritic cells.
Exactly. So this was one experiment that we did in our previous publication, showing that when we reprogram fibroblasts or cancer cells in vitro and inject them into the tumor, we see more potent effects by cancer cells since they bear the endogenous tumor antigens they can then present to the T cells. However, we did see also very potent effects by fibroblasts when we reprogrammed them, since they can cross-present surrounding tumor antigens to the T cells.
Okay. Thank you. Um,
Hi, Ervin.
Hey,
Really nice talk. Amazing. Really, really, I learned a lot.
thank you so much.
Just a just a question about very naive question. I was wondering, because you showed that you also use your reprogramming, uh, reprogramming to reprogram, if I remember correctly, macrophage as
Yeah.
a control. Yeah. So I was wondering, have you tried to reprogram the macrophage? Because we know that, as we all know that tumor-associated macrophage, tumor microenvironment, in this microenvironment, macrophage plays a very important role. So, do you think that it may be possible to reprogram some like macrophage and then combine with reprogram CDC1 and to give a better outcome of the tumor therapy? Just out of curiosity.
Yeah, I mean, it's a very good question. So, what we are also trying to do is combining different reprogramming systems in our lab. So, I think it would be very interesting to assess what would happen if we combine reprogramming of macrophages and reprogramming of DC1s in the same tumor, if there is some synergy, or it could even lead to maybe some antagonistic effects, since macrophages can also adapt anti-inflammatory phenotype. In this case, PO1 and CBP alpha has been mostly shown to drive a pro-inflammatory phenotype with secretion of also pro-inflammatory cytokines, if I remember correctly, and chemokines. However, this needs to be still, I think, investigated. So I think it's a very good question that we could tackle.
And I have a second question. I'm sorry. This is a very, very naive question. Just I'm not an expert of a tumor in study, but I was wondering any reason you compare 2D and 3D? Because I feel like 3D is more relevant to our body, right? Is any
Exactly.
reason why you compare this? Because 2D may be more easy to carry on your research or better to study.
So for us, that's more of a question regarding the reprogramming itself. So from the
Mhm.
perspective of a cell reprogrammer, we need to ensure that we can use reprogramming that was established in 2D as well in 3D. And that's why we also wanted to compare it to the 2D system, whether we can use the same transcription factor combination as well in 3D, and whether they will acquire the same phenotype as well in 3D, since we wanted to use this as a gene therapy in, of course, 3D tumors, which are a whole tissue around to confirm that this can happen. And since of course it's easier to model tumors in 3D with spheroids than to establish all these models in vivo, we went with a spheroid model to test 3D reprogramming and also confirm this in xenografts than in mice in individual selected models.
I see. Thank you so much.
You're welcome.
so we have a question for Patrick. He's asking about the specificity of the viral vectors, if they're only specific for tumor cells, or it can also be turned into, or it can also like be acquired by surrounding cells from the tumor.
Yeah, that's a very good question. And that's also a question we hear a lot. So the viral vectors that we are using are just common viral vectors that are not specific to a certain type of cell yet. Maybe in the future we can develop more targeted vectors, but so far we are going the intratumoral route, just injecting the vectors and then hitting probably not only tumor cells, but also other cells. It might be also fibroblasts, stromal cells, also some immune cells might be hit. And we are also investigating now which cells actually do get hit by the vector and whether we can reprogram, for example, immune cells such as macrophages, which are usually a lot in the tumor as well. It depends on the tumor type, of course, and whether they can acquire also a pro-inflammatory CDC1 phenotype then, whether this is actually synergistic then with the cancer cell reprogramming. I hope this answers your question.
Yeah, thank you very much. I don't think there's are more questions. So thank you everyone for joining today. Please follow our social network, and I will follow your work, Ervin and Pfang. I'm here in the chat box. I put like our social network, and if you want to join our email list, so you can know about our future seminar. So thank you very much, Ervin, you, and everyone to join today. Congratulations.
For inviting.
Uh,
Bye-bye.
Thank you so much.